Hello everyone, I am Suvarna Venkatrao Gaikwad, working as Assistant Professor in Department of Botany of Dada Patil Mahavidyalaya, Karjat, District, Ahmednagar. First of all, I welcome you all in the new topic of T.Y.B.Sc. paper 3rd, that is Genetics and Evolution. In previous session, we have seen the principles of inheritance, in that law of dominance, law of segregation and law of independent assortment we have seen. In today's session, we are going to discuss about incomplete dominance, then codominance, difference between incomplete dominance and codominance, then backcross, and finally test cross. Let's start. So the first point is, what is incomplete dominance? Before we emphasize on incomplete dominance, let's revise the law of dominance. So according to Mendel, law of dominance states that when two homozygous individuals are crossed together, only one character appear in F1 generation, and that character is called as dominant character, and the other character which is not expressed is called as recessive character. So this is law of dominance, but I have already told you that there are some exceptions such as incomplete dominance and codominance. In that incomplete dominance, so see, incomplete dominance and codominance, both they do not follow Mendelian inheritance. Okay, according to Mendelism, the two alleles they segregate independently, and also inheritance of one character is independent on another character. But here in incomplete dominance, out of two alleles, neither allele is dominant and neither allele is recessive. And when the two alleles are in heterozygous condition, they produce a different phenotype. That means the two characters they blend together or they mix together. Here in this example, we have seen there is a red color of flower and white color of flower. But when they are in heterozygous condition, F1 generation or the heterozygous, it doesn't produce red or white variety. It produces new phenotype, that is the pink variety. So this incomplete dominance is common among two plants. The first plant is 4 o'clock plant, that is Mirabilis jalpa, and the second plant is snapdragon plant. The scientific name of snapdragon plant is Antirrhinum majus. So when a cross made between homozygous white flowered plant, the genotype is CW, made with homozygous red flower plant. The genotype is CR. So in F1 generation, we get CR and CW. So this CR/CW is in heterozygous condition. This is similar to Mendelian inheritance, but the difference is in phenotype. If we look at the color of flower is not red and not white. So this is pink variety. How can this is possible? When these F1 generation offspring undergo selfing, then in F2 generation, these plant produces a red color, white color and the pink color. The three phenotypes are in the ratio 1 is to 2 is to 1. That is CR is in red color, CW is in white color, and the two offspring, that is CW, these are in heterozygous condition and they will produce pink color. So how can this pink color appear in F2 generation or in heterozygous condition? The reason is in incomplete dominance, the dominant allele failed to express completely. That is, in this case, the dominant allele capital R failed to express completely. That means it have to suppress the expression of recessive gene, but it fails to express, it fails to suppress, so it fails to suppress completely or express completely, so the term is called as incomplete dominance. Okay, so when we compare this incomplete dominance with the complete dominance, in complete dominance, dominant allele completely masks the expression of recessive gene. That means in complete dominance, when we cross red and white variety, in F1 generation we get all red plants. That is according to Mendelism. But in incomplete dominance, as the incomplete dominant alleles, they do not follow Mendelian inheritance, neither allele is dominant. So capital R fails to express completely or properly, so it cannot suppress the expression of recessive gene, that is small r, so that in F1 generation or in heterozygous condition they will produce a different phenotype, that is pink variety. Okay. So, I hope you will understand the incomplete dominance. The next term is codominance. So, the term indicates co means together. It means out of two alleles or the two genes, they both are expressed together in heterozygous condition. For example, if we cross between red flower with R genotype and the white flower with the W genotype, then in F1 generation RW, they will produce both characters, that is white flower with the red dots. Here in this picture we have seen both colors of the flower, that is white color also and the red color also. That means both alleles they express together. So this is called as codominance. Okay. So this codominance is also present in human beings. For example, ABO blood group system. As we all know that in human beings there are blood groups such as A, B, O, etc. So whenever a cross is made between a blood type A, if a parent having blood type A crossed with the blood type B, in next generation the children having blood groups such as it may be A or B or AB or O. That means the two alleles or the three alleles they inherited are expressed together, which means the term codominance. Okay, and if we compare the incomplete dominance with the codominance, the basic difference is that in incomplete dominance, neither allele is completely dominant nor recessive. That means in this picture, out of red and white, the F1 generation will produce the pink flower. Okay, and in codominance, the allele will both be completely expressed. Means that the flower will show both red and white characters in heterozygous condition. So this is the basic difference between incomplete dominance and codominance. Okay. So next we will move towards the back cross. As the name suggests, cross between F1 hybrid individuals with any one of its parents is called as back cross. As we know that there are two types of parents, one is the dominant and another is the recessive. So back cross also of two types. One is the dominant back cross and second is the recessive back cross. Let's see one by one. The first is the dominant back cross. This is an example of monohybrid cross. So dominant back cross is when a cross takes between F1 hybrid individuals and its dominant parents. We are taking an example with a simple example such as tall and dark plant. If a pure tall pea plant crosses with the pure dwarf pea plant, in F1 generation, we get all hybrid tall plants. Okay, and when these F1 generation again crossed with the dominant parent, that is F1 generation is the heterozygous tall, that is capital T and small t, when crosses with the dominant parent, that is capital T capital T. So in F2 generation all offsprings will be tall. Similarly, when we take an example of dihybrid cross, if a pure yellow round pea plant cross with the pure green wrinkled pea plant in F1 generation, we get all hybrid yellow round plants. And when this F1 generation they crossed with the yellow round plant, that is dominant parent, in F2 generation we get all 100% yellow round plants. That means in dominant back cross, the result of dominant back cross is we get all the offsprings of dominant type. Okay. The next is the recessive backcross. Recessive backcross, the name suggests cross between F1 hybrid individuals with its recessive parents. Okay? Recessive backcross is also called as test cross because it is used to test whether F1 plant is homozygous or heterozygous. That means if the offspring of the test cross show dominant character, individual being tested is homozygous dominant, and if test cross yields offspring of which 50% show dominant character and 50% show recessive character, individual being tested are heterozygous. This helps in plant breeding experiments. Okay. Let us take an example of recessive backcross with the monohybrid traits. When we cross pure tall plant with the pure dwarf, in F1 generation we get hybrid tall as usual. But when hybrid tall variety get crossed with the recessive parent, that is small t, in F2 generation we get tall and dwarf variety with 50-50 percentage. That is 50% tall and 50% dwarf. That is F2 offspring makes a ratio 1:1. Okay. And if we take an example of dihybrid cross, that is dihybrid test cross, when a pure yellow round pea plant crossed with the pure green wrinkled pea plant in F1 generation, we get hybrid yellow round as usual, and when this hybrid yellow round get crossed with the recessive parent, that is green wrinkled, in F2 generation we get all possible number of phenotypes such as yellow round, yellow wrinkled, green round, green wrinkled. These are in 1:1:1:1 proportion. That means as a result of test cross in F2 generation, when we get all the dominant offspring, that means the offsprings are homozygous, and when we get the F2 offsprings out of which 50% are dominant and 50% are recessive, then these are heterozygous in nature. So what is the significance of this test cross? So the most important significance of test cross is it helps to determine the unknown genotype of an individual, means whether it is homozygous or heterozygous. Second significance, it is used in plant breeding to improve the crop varieties. Next, it is an easier and quicker method of obtaining a desirable trait in a pure homozygous condition. Okay, so if we want a pure of a particular desirable trait in a pure condition, then we can follow backcross. Then it is also used frequently in hybridization experiments, and also if you want to increase the number of characters of parents into offspring, then also backcross can be used. And these are some of the references I have been used. And thanking you. I hope you have understand some of the basics of test cross and incomplete dominance. Next we will meet in the new topic of T.Y.B.Sc. paper. Thank you.